as actually from Dr. McLaughlin's lecture, that you can see that Dr. Nelson's lecture is going to follow very nicely. We are really honored to have him here tonight as our guest lecturer. Dr. Charles Nelson is a professor at Harvard University, and he received his Ph.D. in developmental and child psychology from the University of Kansas, after which he was a postdoc at the University of Minnesota. He then joined the faculty at Purdue University in 1984, and the University of Minnesota in 1986, and then he moved to Harvard in 2005. Now, Dr. Nelson's research interest focuses on the effects of early experience on brain and behavioral development, particularly in the areas of memory and facial recognition. He studies typical and atypical developing children and children at risk for neurodevelopmental disorders, such as in cases of autism. And as you'll see, he uses state-of-the-art behavioral as well as different forms of brain imaging tools in his research. Now, his impact on the field is highly recognized, as he has chaired the John D. and Catherine T. MacArthur Foundation Research Network on Early Experience and Brain Development, and he has served on the National Academy of Science panel that wrote, quote, "From neurons to neighborhoods," a phrase that's commonly used nowadays. He recently appeared on the Charlie Rose Show, where he talked about the neurobiology of parenting. So in short, Dr. Nelson is really one of the most influential senior scholars in the field of developmental cognitive neuroscience, and we are so honored that he has taken the time out to be with us here tonight.
Thank you very much. Well, it's a great honor to be here. I should add that I lived in Minnesota for many years, and in the winter we had an expression that there was no such thing as cold weather, just bad clothing. And the analogy here is there's no such thing as having too many slides, you just talk too slow. So you'll see as we go along that we're going to get through a lot of material. It's also 11:30 for me, so I may take a small nap in the middle of my talk. So the first 5 or 10 minutes will be a background into neuroscience and how brains get built over time. And you'll see a few things that will make you smile, I think, and then we're going to dive right into some extraordinarily depressing content. So here's the outline of what I'm going to do. I'm going to begin by talking about the role of experience in brain development. I'm going to talk a little bit about critical periods and what we mean by them, and then the effects of early profound deprivation on development. So just by way of background, for those of you who don't have training in neuroscience, our genes specify the properties of neurons and neural connections to different degrees, in different pathways and at different levels of processing. However, because many aspects of the individual's world are unpredictable, the circuitry of the brain relies on experience to customize the connections to serve the needs of the individual, which is how we can explain how no two brains are alike. And I was giving an example earlier today in Kate's lab that in one line of work we have a pair of identical twins who have the same rare genetic mutation. It's a MECP2 mutation that leads to Rett syndrome, and yet the girls have different phenotypes. So the background genetics is identical, the genetic mutation is identical, but the phenotype is different. So experience shapes these neural connections and interactions, but always within the constraints imposed by genetics. We like to think that some experiences are or should be universal to all members of the species, and Kate talked about this in the context of depriving kittens of light, for example. So sensory input and caregiving help ensure survival. But others may optimize development. So for example, having a sensitive or consistent caregiver, or exposure to complex or age-appropriate language stimulation, or age-appropriate cognitive stimulation. In many cases, both classes of experience have to occur during a narrow window of time, what we call sometimes a critical period or a sensitive period, for development to proceed along a typical developmental trajectory. So we think of sensitive periods as defined as a point in time when the brain is particularly responsive to experience in the form of patterns of activity. But a critical period really represents a special class of sensitive periods that involve irreversible changes in brain function. In other words, if the presence or absence of the experience results in irreversible change, we should be thinking of the term critical period. Otherwise, probably the term sensitive period is more appropriate. For the rest of my talk, I'll probably revert to critical period, but in most cases, I really mean sensitive period. And so I borrow this from my colleague Takao Hench, where you can see what we're talking about here. So we have genes in utero, and then experience and the environment in hinge on the developing brain at these critical periods of development, we can see that there are different critical periods for different domains, and even within a domain, there are different critical periods. So even within the domain of language, there are, we'll think of them as subcritical periods, and then we have behavior on the other end. There are two final points to note. One is that experience cuts both ways. So if a child is exposed to adverse experiences during a critical period of development, infection or maltreatment, or deprived of an expectable experience, nutrition and caregiving, brain development can be undermined. Brain plasticity changes with age. So in some domains, change is possible throughout the lifespan. So learning and memory would be an example of that. And I sometimes give the example of my 90-year-old mother who uses the GPS, a cell phone, a computer, and an iPad, not without calling me 10 times a day to say that something's not working and what's going on. But the fact is she's managed to master this technology. But there are others where change is really not easily accomplished. We don't learn to see better as we get older. So this principle really is illustrated in the next couple of slides. So here's the depressing slide. The ability to change the brain decreases over time. So this is what happens to normal brain plasticity influenced by experience. And you see there's this point of diminishing returns as we go down here. And the reason for that, in part, is the physiologic effort required to enhance neural connections increases dramatically. And so the sheer effort involved in making neurons more plastic gets to the point where it gets harder and harder to do that. And you notice the crossing over point is right about here. So you can figure out where you fit on this plot. Now the next thing is, this is an example of the advantage of acquiring a skill during a sensitive period. So this is, believe it or not, me teaching my 2.5-year-old son to play squash. In case you were confused. This now is 25 years later. This is a movie. This is me clenching my side. This is my son, and this is a doubles squash, and this is another father and son team. And you'll see what happens, and you'll see my point in just a minute.
Six, 5.
My son. That's my son there. And you'll notice how the fathers are really being very supportive of their sons by standing around watching their sons do all the work. Some of you have seen the pictures of Tiger Woods learning to swing a golf club when he was 3. So the point is that I had played squash many years more than my son, but he learned squash at a much younger age. So what I'm going to talk about now is what happens to the brain when there's a profound deviation in the expectable environment. Specifically, what happens to children when they fail to receive stimulation necessary to foster healthy brain development. So Kate went through a little bit of this, and I can probably speed up through some of this. Neglect is the most common form of child maltreatment in the United States. A particularly egregious form of neglect is being raised in conditions of profound psychosocial deprivation. And this is common among the more than 8 million children around the world who are being raised in institutions, that there are more than 140 million parentless children around the world. And more recently, we've come to realize that there are 61 million left behind children in China. These are children whose parents have left them to go find work, and often leaving them with a relative, a neighbor, or often nobody. So this is a big public health issue. So let me set the stage for this. It's 1997. Bill and Hillary Clinton have a conference at the White House on early brain development. And Patricia Kuhl, who's at the University of Washington, was one of the members of that meeting. The same year, the MacArthur Foundation approached me about establishing a research network on early experience and brain development. And in 1999, as an outgrowth of this network, we launched this Bucharest Early Intervention Project. And these are some of the members of the group. This is 2002. Nathan Fox and Charlie Zena are my 2 collaborators on this project. And I won't go through the others, but this is Jack Shonkoff, who authored the Neurons to Neighborhood project. So the goal of this project was to examine the effects of institutionalization on the brain and behavioral development of young children, to determine if an intervention, in our case foster care, high-quality foster care, could ameliorate the negative sequelae of being raised in an institution. And the policy goal was to improve the welfare of children in Romania by establishing foster care as an alternative to institutionalization. So let me get into some background now, and we're going to get into some sobering videos in just a minute. So this is Nicolae Ceausescu. He ran the country from 1966 to 1989. He had this idea that he could increase his power if he increased the population. So he set into motion a series of decrees or laws that were designed to increase the population. So he established the menstrual police. These are state gynecologists who conducted monthly checks of women of childbearing age who had not had at least 5 kids to make sure they hadn't had an abortion, that they weren't using birth control, things like that. He established a celibacy tax. Families got a stipend if they had more than 2 kids, but they were taxed if they had fewer than 5. I recall some presidential candidate once referred to this as voodoo economics. So the fact is, you were taxed if you had fewer than 5 kids. And then he outlawed all contraception and abortion. So the result of this was child abandonment became a national disaster. Families couldn't afford to keep their kids. They were encouraged to turn them over to the state. In fact, as a communist country, Ceausescu basically implored the state, the people in the country, to turn their kids over to the state, and he conveniently built state-run institutions to warehouse these kids. So, December 1989, there's a coup. Ceausescu was overthrown in this coup, and a month later, the Western media floods into the country and discovers more than 170,000 kids languishing in state-run institutions. Poverty was often given as the #1 reason for child abandonment, but there are others. The international media brought the plight of these kids to the attention of the world, and it turns out that large numbers of these children started to be adopted into homes in the United States, in Great Britain, and in Canada. But many of the families who adopted these kids were not prepared for what they had in store for them, because many of these kids had significant developmental problems. But the assumption was that they were deprived of psychosocial stimulation, and if they were simply brought into a loving home, that would cure everything. So in 1990, when the media came in, this is one of the things they found. This takes a second to load. I'm going to let this run without the soundtrack. For some reason, the soundtrack is out of sync. So now at this point, when we go to color in just a minute, this is post-revolution. So this is now January of 1990, although you'll see no difference. And so this gives you a feel for the kinds of places that these kids were growing up in. The mortality rate in many of these institutions would approach 50%. So 10 years later, this is what we found. So this is a room of toddlers, mostly 18 to 24-month-old kids. And I want you to start to notice some behavior. And this is sort of the housing situation where the babies are all lined up in cribs, maybe 10, 15 cribs in a room. So a couple of things to start noticing. First, watch this little girl here. And notice the sheer number of kids. And notice even the kids who are standing are starting to do a little bit of rocking back and forth. Now you notice there's 2 kids there rocking. There's 1 caregiver in this room with these kids. Now, see, 1, 2, 3, there's another one over here rocking. This one's standing back and forth. Notice the caregiver's not really doing anything. She's just really standing there. Now we have more rocking. And these stereotypies are common. This little girl, by the way, we've been tracking until she's 16 years old. So just notice the high rate of stereotypies that you're seeing. The fact that kids this age are not playing with one another at all. It's completely solitary play. So I don't have to do this because Kate did this. These are the reasons why institutional care is bad for the brain. We've known for many decades that children raised in institutions that are at very high risk for a variety of cognitive, social, and behavioral problems, disturbances in social relatedness and attachment, externalizing behavior problems like conduct disorder, inattention hyperactivity, deficits in IQ and executive functions, and a syndrome that mimics autism. I should add that as an autism researcher, roughly 5% of the kids in our study meet criteria for autism, and in another large-scale study in the UK, it's 10%. And then growth stunting. So here's an example of growth stunting. So I want you to be thinking how old these kids are and if they're boys or girls. And this is a 17-year-old girl, and this is a 14-year-old girl. So kids lose about a month of linear growth for about every month they spend in an institution. And so they don't grow. And it's psychosocial dwarfism, and it's not due generally to malnutrition, but rather we think the pituitary gland is not secreting growth hormone at night, and as a result, the kids aren't growing. And it's a classic sign of neglect. What we did is we designed the first ever randomized controlled trial of foster care as an intervention for the social deprivation associated with institutionalization. We screened, when I say we, this is a former colleague of mine, Dana Johnson, screened more than 180 babies to make sure that we weren't going to include kids with obvious genetic, chromosome, neurological problems, fetal alcohol syndrome, and we landed on 136 kids that we thought at the time looked healthy. And they ranged in age between 6 and 31 months, and when we did an extensive baseline assessment on them, they're averaged about 20 months of age. 68 of these kids were randomly assigned to remain in the institution, and 68 were randomly assigned to foster care. And we had another sample of 72 kids who lived with their families and had never been in an institution. And we tracked these kids comprehensively at 9, 18, 30, and 42 months, and then at 54 months. And around now, I was saying, I'm done going to Romania, and it's time to stop the study. But then I found some money, and we saw them again at 8, and I said, I'm done going to Romania, and we're going to stop the study. But then I found more money, and we saw them again at 12, and then you know where this is going. And once again, I said, we're done with this, and we'll complete a 16-year follow-up by December. So we have seen the same group of kids for 16 years now. The three of us come from very different backgrounds, and so we'd have discussions about what we thought was most important to study. And this is how it was resolved. There's really nothing we're not studying at this point, and we keep adding more things over time. And that was a good way for three good friends and colleagues to work out their differences by just going along with everything else that everyone else says. So let's now briefly spend some time on the ethical issues of this study, because I think the minute I said we randomly assigned half to foster care and half to remain in the institution, some of you were wondering about that. So we have informed consent from all the US universities. We informed consent from all the local authorities and the caregivers, in many cases, in Romania. We could do random assignment because it turned out there was no foster care when we started, no government foster care. And we had to build our own. So we did extensive advertising, and for looking for good foster care parents, we then screened them, a psychiatrist, psychologist, social worker, and after all was said and done, we could only find 58 families that we thought would be good foster care families. So we placed 68 kids in those 58 families by keeping siblings together, which was another ethical requirement. So the point here is we were not withholding treatment. There was not enough treatment to go around. That's the way to think of this. Policy of non-interference, which meant that if kids had the opportunity to change their group assignment as time went by, as the vast majority did, we didn't get in the way of that. So if the authorities came in and said, Johnny, who's in the institution, should now be reunited with their biological parents, they went to their biological parents. In fact, by the time kids were 12, there were only 20 to 24 kids still living in an institution. Once the efficacy of the intervention was established, we faced an ethical dilemma, and the term often is used is clinical equipoise. If we obviously knew the treatment group was greatly advantaged over the non-treatment group, then what you would do in a standard clinical trial is you would give everyone the treatment, or you'd stop the study. Well, we couldn't give everyone the treatment because there wasn't enough treatment to go around. And if we stopped the study, all the kids in the institution would go back, all the kids in foster care would go back to the institution. So what we did instead is we worked closely with the US ambassador to Romania, and we called a national press conference, and we announced our findings to the government and to the media. And basically, we shamed them. And we said, look, we were told before this by many professionals that the study was a waste of time, that the kids in institutions look like that because they're defective. It comes from the Soviet principle of defectology. So we said, if that's true, then if we take them out of the institution and put them into a home, they won't get any better. But of course, that wasn't what we found. Within a year, the government passed legislation forbidding the institutionalization of any kid under 2, and they started government foster care. So they obviously heard what happened at the press conference. It's fine print, but we and others have written a great deal about the ethics of this work. In fact, a paper we published in Science in 2007 was accompanied by a commentary that was actually longer than our scientific article, discussing just the ethics of this work. High-quality foster care. Families received a stipend equivalent to the average income in Romania at the time. So this was not like foster care that you commonly see in the United States. The parents were paid a living wage. One parent had to stay at home. They had to be licensed. Close monitoring. Social workers visited the families every 10 days. Every 7 days they met with us to sort of troubleshoot things. We provided all material support. We had a 24-hour on-call pediatrician. And that's sort of the nuts and bolts. And again, we've written a lot about this. But the point is this was very good foster care. So the findings, unless I state otherwise, everything I'm going to talk about adopts what we call an intent to treat design. So in a clinical trial, to avoid sample bias, let's say I'm doing a study of diabetes and everyone in these 2 panels has diabetes, and I give you this wonder drug and you don't get this wonder drug, and half of you get sick and have to stop taking the drug, but the other half of you, your diabetes is cured. If I just analyze the data based on the half that had a good response to treatment, I have a biased sample. It didn't include the half that didn't, that got sick. So the data are analyzed based on the group you were assigned to at the beginning of the study, independent of where you might be living later on. Cognitive development, all right? So baseline, before randomization, Bayley scale, mental development index, average IQ would be 100. So the never institutionalized group is exactly where you expect them, the population mean of about 100. This is the IQ equivalent of the kids in the institution, 64, which is, of course, profoundly low. Foster care, 30, 42, 54 months. In every case, the kids in foster care have higher IQs than the kids who are in the institutional group. But this is the most important slide. This is the timing effect. If we sort the kids in foster care based on age at placement, the kids placed after 2 have markedly lower IQs than the kids placed before 2. This effect persists through 12 years of age. If you look here for full-scale IQ at 12, we still see an intervention effect. Even though the intervention was to build relationships and facilitate attachment, we still see a boost in IQ, which was not what we expected, but we saw very little fade out. But here's an interesting point that we've just sort of started thinking about. Not everyone responded the same. These are the kids in foster care, and it turns out we have one group, the kids in red, who, 23 of them, who consistently over the first 12 years of life have a very high IQ. But there's this other group here that had this slowly eroding IQ. If you look at the kids in the institutional group, you see 3 patterns. There is a small subset right here of increasing. There's 12 of these kids who over time, their IQ is actually improving, whereas the other groups are eroding. And by 16, we have a number of kids now who have IQs below 50. What predicted your group membership was attachment, age of placement, and number of placement disruptions. So this is my summary. Attachment lays the groundwork for all subsequent relationships. Let me just cut right to the chase. If you look at baseline, the percentage of kids with a secure attachment in the never institutional group is about 70%. But in the institutional group, the percentage of kids with an insecure attachment is about 70%. If you look at foster care, the percentage of kids with a secure attachment placed before 22 months is about 60 to 70%. The percentage of kids placed after 22 months is 60 or 70% who have an insecure attachment. Then we did this task called Stranger at the Door. We arranged for the caregiver to let their child answer the door. The stranger knocks on the door and simply says to the child, "Come with me, I have something to show you." And all we're measuring is whether the child walks off with the stranger. 54 months, what you can see is more than 75% of the kids in the institutional group walk off with the stranger. It's cut in half for the kids in foster care, and turns out there was one in the community sample that we've been monitoring. The other thing about attachment, though, is that we think it's a building block for subsequent relationships. So when the kids turned 8, we started looking at peer relationships. And by teacher ratings, the kids in the institutional group are rated as having poorer social skills. But the other thing we did is we created a contrived situation where we brought in a kid from the community to play with a kid in the study, either foster care or institutional group. And we arranged toys and stuff like that, and we just observed how they interacted. And that's what I'm going to show you here. So the boy in the white is a kid who's been in the institution since he's been a baby. He's now 8. And this is a kid from the community. They've never met, so they're going to interact with some toys. And this is one of our psychologists observing this. So first he starts to bite that boy's hand. His language, you don't need to speak Romanian to know his language is delayed. So the point is that these children don't know how to interact with peers. And the other thing that happens is that the other child who might initially make overtures to playing starts to back off and thinks this doesn't feel right. So attachment is compromised, peer relations is compromised. Mental health. At 12, nearly 40% of the kids who have ever been in the institution, foster care or institutional group, meet criteria for some disorder. Internalizing isn't so different between having been in an institution versus not. But if you look at externalizing and ADHD in particular, as Kate pointed out, there's a very high prevalence of ADHD, and it is unaffected by the intervention, as you can see here. This is actually not different. So it's hovering at around 20%. The other thing that's worth noting is callous unemotional traits. So this is maybe a precursor to psychopathy, may have its roots in disturbed attachment relationships. If you look here, for girls and boys, the kids in the care as usual group have a very high rate of callous unemotional traits, but yet it plummets for the kids in foster care. Now, this gender difference is not surprising. Boys usually are higher in this than girls. But you notice we can pretty much wipe it out by putting kids into families. Let's turn to the brain. So we assume that the behavioral phenotype of the institutionalized child reflects alterations in underlying neural substrate. So we turn to EEG and MRI. And so let me just summarize the EEG findings first. Compared to the community controls, the institutionalized kids have lower levels of brain activity across different brain regions and different frequency bands. So let me show you what this means. This is at baseline. We sample brain activity by placing sensors on top of the head. We can color code it based on is it more or less brain activity. And if you look here for the institutional group and here for the never institutional group, if you're looking at the top down, so here's the nose, left ear, right ear, look at the amount of brain activity in the never institutional group sitting over the frontal cortex and what's lacking in the institutional group. You go to foster care. Care as usual, the kids in the institutional group, the kids placed in foster care after 2, they're identical. Kids placed in foster care before 2, and the kids who've never been institutionalized, and they're identical. So like we saw for IQ, we see an inflection point at around the age of 2 years. And I think I took it out, but we see the same thing at age 12. Kate talked a little bit about this. Here's our MRI work. Briefly, we see a dramatic reduction in gray matter for the institutionalized kids and the foster care kids. There's no difference here, so there's no intervention effect for gray matter. Now, Kate was generous in saying that it may be the loss of synapses and dendrites, but it's possible it's also the loss of neurons and cell bodies. So we're not quite sure, but whatever it is very concerning. But for white matter, we see a bump. We see some intervention effect. But one thing you'll notice across all the slides is that you'll notice that when we see intervention effects, we typically don't make these kids look like these kids. We just make them look much better than these kids. So we're improving them, but we're not normalizing them. So MRI revealed a reduction in gray matter and white matter with partial remediation of white matter. I took out my diffusion tensor imaging slides, but we showed that some white matter tracts, the fibers that connect different parts of the brain, recovered with the intervention, but others did not. So much like we see in behavior, we see remediation of some fiber tracts, but not other fiber tracts. And this is a very consistent story. Now, the last part is some work that I have to, where are you? I have to embarrass Kate. So looking at stress was not even on the radar for me. And Kate and Margaret Sheridan, who were postdocs at the time, one day barged into my office, one of the few days I was in my office, and said, you need to look at stress responsivity in the kids in Romania. And I said, how are we going to do that? He said, well, just write a grant and then we'll set up a lab there. And I said, okay, so fine, go away, write the grant and let me know when you're done. And hoping that would be the end of it. So, you know, like 5 hours later, they show up with a grant proposal, which I submit to NIH under my name, of course, because then it was my idea, to build a whole autonomic nervous system laboratory in Bucharest. And then Kate and Margaret went off to Bucharest and set this lab up. So these are the data. We know that disruptions in the stress response system are thought to be a central mechanism by which exposure to adverse life events influences human development. And there's evidence to suggest that caregivers play a critical role in regulating the response children have to stress. Early regulation of the stress response by caregivers may have lasting effects on the development of the stress response system. So early on, we've all seen what 2-year-olds are capable of. They're all about emotion, but not so much about emotion regulation. So when little kids are stressed, it falls to us to help them manage that stress. And over time, they regulate their stress, and their hypothalamic pituitary adrenal axis starts to function normally. So the questions here were, how does profound early life psychosocial deprivation influence autonomic nervous system and HPA axis reactivity? Does randomization to high-quality environments foster care mitigate the effects, and does timing matter? So here's what we did. The kids had to deliver a speech about what makes a good friend, and they had to do this in front of 2 teachers they'd never met before. There's a preparatory phase when they're writing the speech, kind of like Kate and I right before we walked in this room when we were writing our talks. The giving the speech, positive and negative feedback. So as I look out, I'm looking at some of you looking not so positive right now, and others less so. And then there's a math test they have to do at the end. And the whole time we're recording a variety of biological measures. So if we just look at change in beats per minute in heart rate, where you expect heart rate to increase when you're giving a speech. I speak from experience. Here's the speech phase. These are the never institutionalized kids here. Baseline heart rate increases 20 beats a minute. Kids in foster care, about 15. Kids in the institutional group, about 10. So they don't show the typical cardiac response when stressed. If you look at cortisol, these are the kids who've never been in the institution, and particularly during the Trier stress test, they show a big bump in their court response. But the foster care kids and the foster care kids and the institutionalized kids show a flat court response. They're not showing the expected increase in cortisol. But when you divide it by placement, these are the kids' court response placed after 24 months, and these are the kids placed before 24 months. And these kids look just like the kids who've never been in the institution. So psychosocial deprivation is associated with a pervasive pattern of a blunted physiological response to stress in both the sympathetic nervous system and the hypothalamic pituitary adrenal axis. Randomization to high-quality foster care mitigates otherwise persistent effects of early deprivation on the functioning of the stress response system. And we know from other work that when this system isn't working correctly for long periods of time, it predisposes you to a host of health problems as well. And earlier age at placement led to a more normalization of the court response. Now, since I went a little faster than I have, close your eyes for just a second. I took these slides out on the flight here, but I have now time to briefly go through them. We want to know what the biological toll of early institutionalization was. And so the way we did that is we measured telomere length. Telomeres are the tip of the chromosome. Think of the plastic cap on the end of your shoelace. And so what this cap does is, with repeated cell divisions, it protects the cell, but over time, it erodes, much the way your plastic cap frays over time. So here's the protective cap. You can see it right here. And adversity accelerates telomere shortening. So we wanted to know, would we see accelerated telomere shortening in our kids? In an initial paper, we reported just that. When the kids were 54 months, the kids in the institutional group had shorter telomeres than the kids without. We've now done this longitudinally. We have data all the way out to here now. And notice that the kids in the institutional group, now first of all, you expect a decline over age, so that's normal. But notice the slopes. This is the institutional group, this is the never institutional group. As importantly, when you ask caregivers to rate their child's health, what we see is a strong association with telomere length. The kids with the shortest telomeres down here were rated as having the poorest health. And right now, we're actually doing blood spots to look at inflammatory markers to see whether we're also seeing other signs of, say, chronic infection that might be due to this level of adversity. So, children raised in institutions during early development demonstrate significant impairments in physical, cognitive, language, social, emotional, and brain development compared to kids who've grown up in families. Insofar as we've been able to look at our data, our model of foster care appears to be very effective in ameliorating many, but not all, of the negative sequelae of institutionalization. So besides ADHD, we seem to be having a minimal effect on executive functions. But keep in mind, the average age at placement into foster care was 20 months. So if we had to do the study over again, which we're looking to do right now, actually, We would definitely want to see very, very little babies, because the question is, would we see more recovery if the kids on average had been placed in the first year of life, for example? It's imperative to find alternatives to institutionalization. I have given Senate testimony and appeared before the WHO and UNICEF, and there's just no way around justifying institutions except for short-term stays under very dire conditions. So when Ebola hit Sierra Leone and Liberia, for the short term, all those kids whose parents died probably needed to be put into a safe place immediately, but you wouldn't want to keep them there for very long. Children already in institutions should spend as little time there as possible, certainly be removed by 2 years. And finally, we should be thinking about the tens of thousands of kids around the world who are under the care of child protection because they're neglected. And I spent a lot of time with judges who hear these cases, and they are in desperate need of the science, but then they have to make that very difficult decision of when to sort of give up on a parent and say it's for the best interest to get this child out of the home and maybe permanently. So I don't want to make light of this, but we want the science to be carried over to those making these important decisions. So let me end here. This is our team right now in Bucharest, and thank you for your time.
Thank you, Dr. Nelson, for that very interesting and provocative talk. We have a few minutes for questions, if you want to just go to the mics. Can't really, hard to see with the lights in my eyes, but you just blew them over. Yeah. Oh, we have one question. I don't know if that person's.
Thank you very much for that. Given the kind of associations coming out right now between autism, spectrum disorders, and mirror neuron development, do you see any discrepancies in mirror neuron development in these psychosocially deprived children?
So that's a good question. We actually did not do a task that we think would activate that system. So we can't say if, for example, if imitation is impaired, and if on our EEG we see mu suppression, since I have a feeling you know what I'm talking about. So we actually didn't look at that. The expectation, though, would be, as it is in autism, that we would see a difference in our kids. But again, it's one of those things that we didn't think to do at the time, but it's a good question.
Over here.
Hi, so my question's about, when you're having your conversations with all the judges and sort of policymakers in the US, have you given them any suggestions of ways we can change our foster care system, maybe low-hanging fruit that can be easily done without lots of money or, huge shifts in policy? Just trying to think of ways of how we can learn lessons from your high-quality foster care and apply it to our own kind of broken system.
Oh, so the brief answer is no. And the
answers.
longer answer is this. So I lecture in the law school on occasion with two colleagues. One is a retired federal court judge, and one is a woman who does international law. And they nail me every time. So they ask that question. And I say, I'm just a scientist. I just collect the data. It's not my responsibility to, for you to help you figure out what to do. And then they say something like, that's bull, right? And then particularly the law students go after me. So I think that a way to start, and this has been done, of course, is every year I and others will spend a day or two with juvenile court judges and social workers who hear these cases, so they understand the data. There's nothing unique about kids growing up in institutions. Any of you in the audience who have ever worked with kids in the US who are neglected, the phenotype is vastly similar, just maybe not as extreme. So in the end, I think that what we try to convey to them is there's a timing issue, and there's the issue of if this persists for too long, so there's the duration and the timing issue. If this goes on for too long, you should assume, I'll give you a personal example. So I'm married to a lawyer who for many years did child protection, headed up big child protection division. And then she switched to juvenile prosecution. And who was she prosecuting? All the kids she dealt with in child protection. And so judges are hearing this, but I'm very sympathetic to how hard these decisions are. And I don't want to make light of that. So that's why I wanted to just say no to the beginning, because we can talk about it, but in the end, they're making these decisions, and they can affect people's lives. But we're going to start a project in Brazil, and there, a lot of kids are abandoned or removed from the home, mostly because of drugs. And trying to rehabilitate those kids with those families fails in the vast majority of cases if in their case, crack is involved. So it's a great question, but I'm just a scientist, and my job is to collect the data.
Thank you.
So my question is, your studies were done in Romania, but, to what extent do you think your findings apply to other populations that have been institutionalized, such as, Native Americans and other First Nation populations?
Excellent question. So we started getting wind that different countries were responding differently to these data. So in South Africa, which has a big problem with kids in institutions, they said, those are Romanian kids. What does that have to do with kids in South Africa? Other parts, even within Africa, like Uganda and other countries, said we should pay close attention. The Lumos Foundation, which was started by J.K. Rowling, Harry Potter, for those of you who are, remember Harry was an orphan. They approached us to launch. They said, listen, would you be interested in replicating this in 3 different countries, a high resource, low resource, and middle resource country, to increase the generalizability of the findings? And that's what we're now doing.
That's a great idea. Which countries, she's asking?
We're almost certain we'll start in Brazil. The other targeted countries, Japan was high on the list, but it's been crossed off. Japan has a huge number of kids in institutions, almost all for child protection cases. Zambia, Colombia, maybe Nicaragua, and in Asia, it could be Malaysia or Indonesia. But we're not sure yet. We first have to raise a lot of money.
One question over here.
Yes, the children in the institution looked extremely malnourished, and I'm wondering if some of their problems were because of being malnourished as well as being neglected and deprived?
Yeah, so that's a great question. One of the many things we failed to think about at the beginning was whether the kids were malnourished. But what we were able to do later is we actually got a hold of the menus of the food being served in the institutions and had a dietitian look at them. And the results were, now we don't know what they actually ate, but at least the food being served was adequate. And so in our study and in others, I don't want to minimize nutrition, but we don't think it's a major player here because we think the kids were being fed. And not an ideal diet, but they were getting the right number of calories and maybe not the right balance of things. But I have to say that in other truly bad institutions, clearly nutrition plays a big role.
It looked like there wasn't really a lot of exposure to any kind of media or games or things like that, any kind of exposure at all. Would that make any difference at all, even if you didn't have a real human, you'd have maybe something virtual or just media or TV even?
Yeah,
anything like that?
That's an interesting question, and I'm going to back into it another way. Play is essential in child development. Play is not facilitated in these kids. As a rule, they don't have toys. The only TVs I've ever seen are in the staff rooms where the staff is watching television. So there's no TVs. I don't know if there's radios. The toys are usually donations from some NGO, but it doesn't look at all like a daycare center. So the extent to which kids are prevented from or inhibited from engaging in play is, I think, a serious issue. And you'll notice those kids all in the room together, not interacting with one another. And whether that's for reasons of developmental delay or disorder, or because that's never been facilitated, we don't know. So play is an important issue here.
you did say that this is similar to neglect in the United States, where we have plenty of media surrounding kids all the time and potentially toys and things.
So I think the difference is that I have to be careful here. Having access to media is no substitution for having access to a social being. And in fact, Sarah, I can't say his name, Dimitri Christakis. That's him. You know, it's shown that kids with media exposure can, videos in particular, suffer the consequences. And Pat Kuhl has demonstrated that kids will not learn language from video or audio material. They need it from a live person. So even having access, I've had a lot of, usually they're undergraduates, who think, oh, what if I develop they're in engineering or computer science. They want to develop an app thinking that's a substitution for a human being. In autism, of course, people are working on robots that they think kids with autism might be more comfortable interacting with. That's fine if you live in a world of robots, but you don't. And so there's still the issue that we're social beings, and there's no substitution for that. And we know how critical that is in development. So I think even if we gave them all the technology at our disposal, the outcome would be minimally different.
Okay, I think with that, to respect everybody's time, because it's just after 9:30. Thank you again for coming, and thank you, Dr. Nelson.